Welcome to the London Futurists podcast. Within the longevity research community, Alex Zhavoronkov is well known for his intense focus. He works 18 hours a day, 365 days a year. He takes no holidays and has no plans to start a family unless he meets a partner who is at least as focused as himself. The subject of this relentless attention is the aging process, how to understand it, arrest it, and reverse it. The primary vehicle for Alex's work is Insilico, a drug discovery company that he founded in 2014, which uses artificial intelligence to identify novel targets and novel molecules for pharmaceutical companies. Insilico now has drugs designed with AI in human clinical trials. Alex, welcome to the London Futurist podcast.
Thank you for having me, Callum and David.
It's a great pleasure to have you here, Alex.
Very happy to be there, and I do remember that I did present at one of the London Futurist events. I think it was more than 10 years ago. Great memory.
Perhaps we could start by you explaining what the state of play is at Insilico, because in February this year, if I'm not mistaken, you got a drug for idiopathic pulmonary fibrosis, IPF, to be the first phase 1 clinical trial of a drug developed from scratch using AI. So where are we now?
So at Insilico, I think we're in the best time of the company existence. So we've never felt better. And for the first time, we do have the room to breathe, so to speak, because we're very well funded and many industry players are using our software. And now we have the ability to develop it even further. And we've managed to progress a lot of preclinical programs closer to the clinic. So now we have internal pipeline of 31 therapeutic programs, 29 of them fully owned, and one has progressed into human clinical trials. And for those of you who are not familiar with drug discovery and how drugs are usually discovered and designed, it's probably very worthwhile to give a very brief tutorial. Here, you just need to pay attention to the numbers and the process. Usually, when we are approaching a disease, we need to first identify the disease hypothesis. So we need to invent a reason why disease is happening. So for example, for Alzheimer's, there are many hypotheses, but none are fully validated. And just recently we saw that a hypothesis brought forward 20 years ago is not valid. So it was brought forward using false information. And a lot of scientists are now debating that. So after you come up with a hypothesis, in the Alzheimer's case, for example, a faulty hypothesis dealing with certain protein plaque accumulation in the brain, and people were trying to target that mechanism and failed many times over. So using those accumulated proteins as protein targets. So very often the target hypothesis itself, when it's wrong, you are failing to find a good target.
And the target, Alex, is usually a protein, isn't it, in most cases?
Usually, most of the time, it is a protein. It could be a biological process, and usually it's more than one protein that's involved in the process. But usually, if you are going the standard FDA, so Food and Drug Administration recommended route, you are looking at one drug, one disease, one target. Mm-hmm. That's classics. That's what most people are going after.
And the part of your company that does this bit, identifying a target, is known as Pandaomics, is that right?
Yes, that's correct. So we do have a software product that helps you come up with a disease hypothesis using multiple approaches, multiple data types. So that's not just one AI model. It's a combination of more than 20 AI models trained on different data types that allow you to, first of all, bring your thoughts together by utilizing prior knowledge from text, but that gives you a little bit more confidence, and also identify completely novel targets by utilizing human biological data from public repositories. We do not use proprietary patient data. We use government-funded projects that are put on public repositories, and it allows us to identify those critical molecular switches within certain diseases that if you switch them off, or on, the disease is either ameliorated or it's cured. There are some curative targets as well. So the first order of business is to identify the link between this critical pathway switch, the target, and the disease. And the important thing to know here is that there are probably over 60,000 targets out there. So depending on how you look at it, you can narrow it down to a few thousand. And out there in the disease codes, you will find over 10,000 diseases that could be targeted.
So there's a lot to play for.
Yes, and usually the disease does not transpire in the same way in two different patients, right? So usually something is different. So you need to find something that works for as many patients as possible, and then also learn how to identify those that respond. Mm-hmm. So that is the job of our platform called Pandaomics. And that's the first step of drug discovery. And what's important to understand about this is that usually is done in the academic circles. So academics work, and also clinicians, clinical researchers work for many years, spending a lot of public funds to come up with a hypothesis on some of those targets. Usually it costs billions of dollars, for Alzheimer's, tens of billions of dollars, and for some cancers to identify some of those targets. And then it fails 95 to 99% of the time. So 1 to 5% success rate. Pretty substantial probability of failure. And also it can take you decades. So that step is the most important step. After this, after you've identified the target and validated it in some preclinical models, so you need to have some mice or some human tissue with the disease to be able to test your hypothesis in order to progress further. So that is called target validation. So that takes also a lot of time. And once you've identified the target and validated it to some extent, you start designing either an antibody that disables that target, or most commonly you start developing a small molecule, so a specific chemistry to go after it.
And this is the part of your company called Chemistry 42, isn't it, that does this part?
Yes, that's correct. So we decided to go end to end. That part from target discovery, so when your target is discovered, so it's called target to hit. You first identify a weak binder to the protein of interest, and then you try to improve it. So usually you can do it using high throughput screening. So make this protein, put it in microplates, and then test many compounds, many promising compounds, sometimes thousands, to see if it binds. So that's the classical approach. The way AI looks at it, it allows you to look at the protein, see where the possible binding pockets are, and then imagine novel molecules with the desired properties that might be binding to this protein. And that allows you to synthesize just maybe 10, 15 molecules, test them, and see if those absolutely novel molecules work or not in an experimental system.
What kind of AI are you using to do that? I think I've heard you say that this part is done by generative adversarial networks. Is that right?
That's correct. So we use a multitude of generative algorithms. So those could be genetic algorithms. Most of the approaches are GAN or GAN-like, so generative adversarial networks. Some are transformer neural networks. So we have a zoo of over 40 validated models. So validated meaning that we've used this model to generate novel chemistry. And after that, we synthesized that chemistry and tested this chemistry. So sometimes to come up with an AI model takes you only a few months, but then it takes you a year to validate. Unlike with pictures, where you generate beautiful art and you see that your generation conditions are met in that art. So in chemistry, it just doesn't work like that. You don't have a trained human who can tell you that this molecule is going to work. Otherwise, they would have drawn this molecule themselves, and very often they do. So in our case, we have over 40 trained models that compete with each other to design a small molecule with the desired properties. And that process usually takes to go from target to a preclinical candidate. That's where you validated many molecules and optimize those molecules in many preclinical models. So you showed that the molecule works in animals. So it cures cancer, for example, in certain animal model, and also in vitro, so in petri dish using human cells. So that process usually takes about 3 years. Very often it takes 4 or 5 years, depending on how the company is progressing. So Our platform demonstrated that you can do this, including target discovery, in under 18 months with our antifibrotic. So new target, new molecule, under 18 months to preclinical candidate. And we showed that we can now also go into human clinical trials with this. A novel combo, novel target, novel molecule. The target has never been in the clinical trials before. It has not been validated. So we validated it, generated small molecules with the desired properties, also using another piece of our puzzle called Insilico.
Before we get into that, Alex, what you've just described is like a tenfold improvement or more. And these are very large sums of money and time we're talking about. Why on earth isn't the entire pharmaceutical industry now doing this and abandoning their old ways of doing things? We'll be right back after a quick break.
The People in Performance podcast offers insights into the strategic competencies, behaviors, and results needed to increase the performance of employees and leaders. The show looks at all areas of the world of work, including attraction and recruitment, onboarding, training, and succession planning. Tune in to get a better handle on the stages of workforce planning you need to master in order to guarantee long-term success in a rapidly changing world of work. New episodes drop each week. Check out the latest People in Performance episodes on your podcast platform of choice. Follow us on socials and read the latest articles at pppod.com.
Well, they are trying. It takes time to transition. So all of them have AI groups. All of them are trying to utilize those algorithms. Many of them do use them. It just, for a large behemoth, it takes a lot of time to change their ways. Also, it's very important to understand the statistics. That's what I'm saying. Usually, the probabilities in this game are more skewed towards failure. So in phase 2, that's where you test efficacy. Even after you tested the molecule in preclinical models, once you go human and you've already spent $1 billion and you are testing the molecule in phase 2 clinical trial to find a blockbuster therapeutic, it fails more often than it succeeds. So usually it's 60% failure rate.
Amazing.
It's a market of failure. So here it's not about the speed up. It's all about the probability of success.
Can you compare two approaches for me, Alex? You've described that the system imagines new molecules that have never existed before, so that these molecules would potentially fit into the right place in proteins and have the right characteristics, and then you need to validate that they are safe. Whereas another approach I've often heard discussed with AI drug discovery is to use the properties of known drugs that weren't particularly successful previously, but they at least are known to be safe. So if your system could validate that these other drugs might fit in instead, then it could speed things up even further. So what's the pros and cons of inventing new drugs versus testing existing drugs, both in silicon?
Absolutely. So what you've discussed is called drug repurposing, taking a known therapeutic and putting it to a new use. We've done that before. We've done that with pharmaceutical drugs and also with nutraceuticals, believe it or not, right? So we tried that on something that not only has a safety profile as a drug, it's also generally recognized as safe, so you can put it on the market. The problem with this approach is that if you are repurposing a therapeutic, usually it belongs to somebody, so somebody owns a patent on it, and you need to go and in-license. And that's a completely different business model that we thought about, but we did not pursue it. We decided to own the intellectual property that we create, and also speed up the search for molecular properties, for molecules with desired properties, and also imagine novel molecules that do not exist in nature. Because those molecules that you can in-license, they're usually still pretty bad, right? So they could have been better. They don't have all the properties you want. They may be moderately safe, but they are not ideal. So we decided not to go that route. The company that is going that route, and the company that your audience should pay attention to, and you might want to interview the founder, Christian Fortney. The company is called BioAge. So they identify targets that already have known drugs that may have dual purpose, that may work in the disease and also in aging. And they go and in-license those drugs. The problem with this approach, again, you don't own the intellectual property. Very often those molecules that you want to repurpose are already generics, so they don't have the patent life. And if you really progress them into clinical trials and succeed, it takes you a few years, it will not be commercially viable. So investors will not give you the money, at least the smart investors. So you need to be extremely careful. And in the ideal case, when you find a therapeutic with long patent life, you found somebody who is willing to license it to you, you are very limited with the number of those that you can license. So usually there is like a handful that are promising, licensable, and do have patent life. So you are very limited. In our approach, I can go after almost any druggable target. And the beauty of our program one, the anti-fibrotic, is that we actually discovered it using aging research. So the way we identified the target, it's not only Pandaomics, it's also a deep neural network which is trained on longitudinal data, so on healthy people, and also people with fibrosis. So we look at the targets that are implicated in aging and disease at the same time. So I call it dual purpose therapeutics. So in this case, you don't really need to do a clinical study for aging. You can do a clinical study for a specific disease. And as a bonus, you are addressing aging. And while you are doing the clinical study, you can also include some data types that you are going to be collecting during the clinical study that will help you answer the question whether the drug works in aging or not. And in that case, it's so much more superior than trying to repurpose older molecules because you can have a commercially viable product. So you have a case for the investors where you have a tangible case of possibly returning their investment with 10x, 50x, 100x return. And you do get the data you need to be able to evaluate whether this therapeutic is working in aging or not, because you are just collecting the same data that you would be collecting in a very comprehensive clinical study. So you can collect proteomics, transcriptomics from all kinds of biological data that changes in time dramatically. You can collect that data and run aging clocks, biological markers of aging on this data. And you would be able to get this question answered, whether your drug that works in a disease also works in aging. To do that, you also need to be extremely safe. So you need to go and do multiple safety studies. So you need to ensure that this drug is okay for chronic conditions, for something that people would take over the entire course of life, right? So you're targeting metformin type of safety or better.
That's very interesting. And I'm sorry, I interrupted you earlier. You were about to tell us about the third phase of your process in clinico.
Yeah, so when we started the company, we thought, okay, where can AI help accelerate drug discovery and development? So we came up with this tool called Pandaomics for target discovery, the tool called Chemistry 42 for small molecule generation, and we also built a tool also reusing some of the models from early stage drug discovery called In Clinico, which looks at the clinical stage programs and predicts whether they are going to succeed in phase 2 clinical trials where you test efficacy. So we imagine that the drug passed phase 1, so it's safe, and we test efficacy. So mostly looking at how implicated the target is in a disease and how heterogeneous the target is in a population. So how often is it very important in a specific disease in as many patients as possible, right? And how many patients. So we try to evaluate those data points, plus many, many others about clinical study design, molecular profile. And we make this prediction whether the drug is going to pass phase 2 to phase 3. And usually when you combine all three engines together, you get the best result. And many of the engines in Clinico are trained independently from our target discovery tools. So they are actually trained on massive data sets of already completed clinical trials that are in the public domain. So we look at many butterfly effects even that influence the success or failure of clinical trials, and our AI is also interpretable. So for every one of our programs, we also apply In Clinico to assess the probability of success and to ensure that it's maximized. And this system can also be used for repurposing, so what David suggested before. So you can actually look at a known drug that's in a clinical study and try to test it against other indications to see if the probability of success is higher there. And some of those indications could be age-associated diseases, or it can even put aging as a disease.
This in clinical system seems remarkable to me. The idea that an AI could look at data in advance and figure out the likelihood of a clinical trial being successful. I can understand how such a system could be trained. Can you say how much has been verified so far? Because presumably to know whether it's actually giving you good forecasts, you need to go all the way to the end of a clinical trial to see whether it was correct or not. Have you got that data yet?
Oh, very smart question, by the way, David. This system is one of the oldest systems that we started developing at Insilico. We actually started from that system because I tried to go into clinical stage portfolio theory to see how we can use AI to combine different clinical stage programs to be able to maximize the chances of success. At that time, I was thinking about cancer and longevity mega funds, right? Thinking about applying the system for that purpose. So we started working on that before any other system. And we realized that retrospective validation doesn't work, right? So if you just take 10% of the clinical studies out and use it as your test set, so you train on 90% and then test it on the 10% that you've taken out, you are going to be still biased. A much better approach is called quasi-prospective validation, where you train all your AI up until 2015, for example, on the entire totality of all the clinical studies that completed on the entire human history before 2015, and then test it on 2015 to 2020, right? So see how it worked on other clinical studies. And even that is not enough to be able to validate. So the ultimate validation comes from truly prospective studies where you make a prediction, you explain how you did it in detail, and you put it online where it's date stamped. And then you wait. And you wait for a year, you wait for 2 years, you wait for 3 years. Sometimes you need to wait for 6 years. And we've done that too. So our first paper hit the archive, so a preprint server with a date stamp, in 2016. And our last paper, a prediction of clinical readouts for a big pharma company, hit the preprint server, ResearchGate in this case, just a couple months ago. So also predicting the clinical trials. So in most cases, the success rate of our system is over 90%. Not all the clinical trials are predictable. So we can predict about 20% of clinical trials that have sufficient data that's available to us in the public domain. But for those 20%, we do very well. What we also did, we did a 1-year pilot with one of the pharmaceutical companies. I'm not going to tell you which one. So they also challenged us for their internal purposes to work with their team to predict 10 trials that they knew are going to complete within this year. So in the beginning of the year, we started the pilot, and 8 of the clinical studies completed by the end of the year. All of them we predicted correctly, and 2, they were postponed, so for different reasons. So later they also completed, and we predicted 1 of them correctly, so we had 1 failure out of 10. The problem is that this pharmaceutical company later, they did the review, they analyzed it, they were amazed, and we got amazing feedback. And we, of course, had bi-weekly meetings with the project teams to show how we do it as well, and to ensure that we just keep in touch. But after the project was completed, they just went quiet. Well, first of all, a couple people quit by that time. You know, pharma is a dynamic industry. And internally, they decided, okay, how are we going to use it? If we are going to use it on all programs, are we going to terminate those phase 2s that are midway?
That's what you would think they should do. I mean, that's the whole point of doing it, isn't it?
They would not trust an outsider to do that. Whatever validation you show them. So right now, if somebody comes to me and says, Alex, your program 1 is likely to fail in phase 2 for fibrosis, and here is why. So I would, of course, look at why, but I think that we are the best in the world in predicting clinical trials outcomes, specifically for those cases. Nobody else has done a study like ours. Should I really listen to those other guys? Well, now it's going to introduce a lot of FUD, fear, uncertainty, and doubt for my own program. So that's what pharma companies are thinking about as well. The biggest fear is that we were told not to do that. We were told to abandon, and we didn't, and we failed, right? And that could be a pretty dark spot. So they would prefer not to know. So who really likes to know are hedge funds and banks who are betting on small and medium type biotechs. And that's where we are making many pilots right now. So we're doing pilots with some of the smartest humans who actually bet money on those outcomes. Large companies.
You've got to find the people with the right incentives.
Yeah, and that's where we're validating. So I think that this is an extremely promising area. And we also use this tool to identify those dual purpose therapeutics to go after aging.
Alex, about your program 1, the IPF trial. You've already said it's accelerated compared to what it would have been otherwise, but how long is it, do you think, before you could have a commercially useful treatment available for people, if all goes well?
So it's a very good question. I think that to answer this question, we need to complete the phase 2 study. And the fastest you can do, no forward-looking statements, not an investment advice. We're not a public company, but I need to make those statements. The earliest you could see the phase 2 data would be late next year, beginning of 2024, from IPF patients. That is, of course, a major inflection point for the company. Even completing phase 1 would be a pretty dramatic inflection point to get to this point. It's very rare, considering the fact that we've identified this target using aging research. We, in my opinion, achieved many world firsts, right? Acceleration, probability of success, dual purpose therapeutic, aging research as a starting point, and aging clock as a starting point. So possibly we'll be able to test this particular target with an aging clock as well. So it's an aging clock derived therapeutic. And it worked so well in many preclinical models that, I have very high confidence that it's going to work. But it's going to take another 1.5 years to see the data in humans from phase 2. And after that, it would take a regular couple years to get through phase 3. So I would say 4 years, yeah.
Well, hopefully you'll come back on the show and tell us how that's gone. An interesting feature of your company is, you started your career in Canada and America. I think you're from Latvia originally, but you studied in Canada and you were very successful in the semiconductor industry in America. But Insilico was set up in Hong Kong, and you're based now in China and then to an increasing degree in the Gulf states. Is there a reason for that geographic shift? Is there something favorable you see in those territories?
Well, nowadays, a lot of people's minds are extremely biased. So whatever you say about China, people perceive negatively because of just negative media pressure. I think that currently the biotechnology industry in China is very similar to IT in early 2000s. Many pharma companies and biotechs are using Chinese CROs, contract research organizations, that allow you to do research without having your own lab. So us as an AI company, we don't want to build massive, gigantic research facilities to compete with big pharma. So we can use CROs, and there are many of them, and most of them, the largest ones and the most influential ones, are in China. After hundreds of billions of dollars of investment on the part of institutional investors, and also on the government side, right? So they really wanted to become the central hub where everybody comes to do the research, just like in the past, people were coming there to do manufacturing. So we can basically follow the Apple model where it's designed anywhere in the world, say designed in the US, made in China. So here you have designed anywhere in the world, say California, but synthesized and tested in vitro models and then in vivo models in China. You can even do the clinical study there as well. And also nowadays they opened up pretty dramatically and accelerated, and you can even do some of the clinical studies faster there too. Usually you would do 2 in parallel in the US and China, just because of the fear that the regulators are not going to accept another country's data for safety and efficacy. So for us, we wanted to accelerate, and we didn't want to build our own lab before we train our AI and we validate. So we had to use those CROs, and being close to those CROs, you really need to also have your own people within those CROs, because you want to ensure that you have your own managers managing those full-time equivalents, so scientists that work for you on your experiments, that you get the best people working for you, that they are extremely motivated, that they do the work right? So quality control. So that's why we also decided to be in Hong Kong, which is kind of, one country, two systems. It has amazing intellectual property protection system. It's a financial mega hub. It's a great stock market here as well. Beautiful place to live. Has a Hong Kong Science Technology Research Park, and it's right next door. Before COVID, it was very easy to operate out of Hong Kong. Afterward, we also needed to set up a site in Shanghai, and we progressed quite a bit. And then I met my chief science officer, who originally started as a chief science officer, amazing big pharma executive, Harvard PhD, 11 years at GSK in neuroscience, so also knows a lot about aging. And then he helped list a company called Medicine, which is a contract research organization, a huge $8 billion at that time contract research organization. Once he saw what we can do with AI, he actually decided to join us. Originally as an advisor and then as the CSO. And now we have a double CEO, so dual CEO system as well, where he became a CEO. And he manages drug discovery. So he uses AI to accelerate and propagate our clinical programs. As a matter of fact, what really frustrates me today is that people in longevity even are against international collaboration. Remember the good old days of Clinton, Obama, and many others, where people were pushing towards globalization and trying to integrate. Nowadays, partly due to aging, because nowadays the economics of aging start to kick in. So what I proposed in the book that I published 10 years ago is happening. So now the major economies are in deep trouble, and not only because of the recent geopolitical events, it's also because of aging. It's a new paradigm. So if you look at the net present value of the unfunded fiscal imbalance in the developed countries, in the US specifically, because it's the beating heart of the global economy, it's dramatic. So when I published the book, it was $300 trillion, right, in net present value in terms of liabilities to the retirees. Nowadays, I just don't want to even do the calculations. So even the national debt is over 30 trillion. When I published the book, it was under 13 trillion. So it's more than doubled. Right now, we're getting to this inflection point in the economy where there needs to be a reset. And I really hope that this reset is not going to deprioritize aging research completely, because people will be striving for survival. But any kind of nationalistic and anti-globalization initiatives are bad for longevity, and specifically bad for biotech. It requires massive collaborations. And a drug discovered in China will be curing Americans, and it will be curing the Europeans. And again, the probability of success of discovering such a drug is very low and requires massive investments. So you have to have an open mind and an open heart when it comes to biotechnology. You can compete on anything else, but biotech is the place where you need to collaborate. And I know that a lot of people are against that. A lot of people look at me negatively. A lot of people assume that I am Russian. I never had a Russian citizenship. They just assume because of the accent and the family name, and are trying to undermine the company using that particular angle, which is pretty bad.
That's a great answer, Alex. You've explained that very well. You referred to one of your frustrations being the resistance to international cooperation. I wonder, are there other things that are slightly frustrating you, preventing your company from bringing out products more quickly? For example, do you need more data? You referred to the access to data from government-curated repositories. Do you need more data? Or do you need more animals in your zoo of AI models? Or do you need to understand chemistry better so that your systems could more accurately and more precisely come up with chemicals that are likely to work?
Regarding the data, we already have all the data we need because we follow the government grants. I integrate $1.5 to $2 trillion worth of grant funding in a database, right? And it's linked to omics repositories, openly accessible clinical data. So AI is actually trained, our AI is trained on dirty data, but it's tested on ultra high quality data sets that we trust. And since my software is actually available to the others, they are doing a lot of validation. I think that right now my biggest problem is ultra highly qualified talent. We're extremely famous, right, in our industry. So a lot of people come to us, but having all the skill sets we need to integrate into Insilico, only a few people can do that. Right now, my biggest problem is talent, and specifically not even AI talent, but drug discovery talent and even business development talent that requires you to be a Swiss Army knife. Basically, I need to find a few more people like me. Actually, one of the reasons why I'm doing a lot of advocacy and outreach is to try to create more people like me, because then you have highly effective managers that can bring together scientists and business people to even build companies like mine. And so far, we've created a few people like that already. But it's not enough, and we need to ensure that there is more talent. So more talent will help a lot. Another thing is, of course, it requires government competence. It's not about the resources. In some regions, we really do need to have specific government support. They need to realize that, okay, there are only a few companies that are at the very top, at this food chain. It's very difficult to get to clinical stage from AI. There are hundreds, thousands of AI companies out there, and many of them are doing massive PR, and some of them are actually led by extremely credible people. But where are the clinical stage assets? And right now, if I were the regulator, if I were the FDA, if I were the NMPA, if I were a regulator in pretty much any country, I would try to focus on us as a case study. I would try to do a deeper dive. Instead of waiting for our submission, I would try to do the inquiry. I would try to create a specific task force to try and do a test case, whether the AI designed and the AI discovered therapeutic could go into clinical studies and succeed, and can we also accelerate clinical development? So that would help a lot. Unfortunately, again, there is a lot of noise in the industry. And again, those geopolitics get in the way of science, right? And very often the countries are prioritizing something, but they are prioritizing something wrong. So we need to ensure that they just get better internal analytics to accelerate the right programs just to test if it's possible. And we are the great test case.
Well, Alex, let's hope you do get that regulatory cooperation, and let's hope you do find that great talent, although, to be honest, I think you're going to find it difficult to replicate yourself. But let's hope you do achieve those things, because what you're doing is very exciting and it's extremely important. And we're very grateful to you for coming to share it with our listeners on the podcast.
Very happy to be here. And again, I think that doing what we do is probably the most altruistic thing anybody can do, because you're creating more life.
So if people are listening to this wondering how they can make a big difference in the world, then actually they should pay more attention to what Insilico Medicine is doing, and more generally looking at ways in which they could help more people to live healthily for longer.
Yeah.
Absolutely. And actually for that, we did create a course. So it's not part of Insilico. It's a volunteer effort. It's called Longevity Medicine course. You can find it at longevity.degree, CME accredited. Very simple, so a physician can take it without any prior biological training. And a few thousand physicians have taken it, where we explain the basic concepts of what aging is, what is out there that might hold promise. And once you, of course, go through this course, your expectations are going to go down, because you realize how long it takes to discover and develop a therapeutic or even repurpose, and how slow this industry is moving. But it's something that is necessary. So I wish somebody gave me this kind of tutorial when I was starting. When I first met David, I was much more optimistic, and Callum, you as well.
We'll definitely put that in the show notes. But thanks again, Alex, for joining us on the podcast.
Thank you.
Thank you.